All right, this lesson is about DNA replication. We're doing topic 2.7. Now, just a reminder, this is only half of topic 2.7. The rest is on transcription translation, and that will happen next lesson. So just the replication dot point so far. All right, DNA replication is the synthesis of new double-stranded DNA molecules. All right, Watson and Crick with Franklin determined the DNA structure in the 1950s, but no one else was really sure of how it worked. So scientists knew it had to replicate, pass on those hereditary traits, but they needed to work with the model, the structure, to determine how it actually did it. Now, DNA replication is part of the cell cycle. It's part of S phase, and the DNA synthesis happens before mitosis, because mitosis is when that nucleus actually splits. So the DNA synthesis has to occur prior to mitosis. And remembering back, what we are trying to do is go from a single double-stranded chromosome to having those sister chromatids so that they can be pulled apart during those mitosis phases. DNA replication has a number of steps, but it is quite simple at its crux. So number one, the DNA's, so the ladder is unzipped. It is then rebuilt from the template strand, and then those two split to go into different cells during mitosis. Right, so the very first step is using an enzyme called helicase. Now, helicase unwinds the DNA at a particular point, and it breaks those hydrogen bonds between the complementary base pairs. Now, it does separate each side of the DNA strand, but it doesn't do it the whole strand at a time. It just does little chunks, and when it's finished that bit, the other part can continue to wind back up again. All right, DNA replication has a second step, obviously, and that is when we have to do that rebuilding. So the open strands act as templates, and then another enzyme, DNA polymerase, brings in free nucleotides that are floating around the nucleus, okay? And it builds that new complementary strand, complementary to the base pairs in the template that are already there. It goes from the 5' end to the 3' end, and DNA polymerase has a number of jobs. So it builds that new strand by bringing in those nucleotides. It can also proofread, and then it also needs to kind of seal up that backbone, so the sugar phosphate backbone. So it's facilitating those hydrogen bonds back between the complementary nucleotides. Now, that third step is all about making sure that everything's good to go, ready for those chromosomes to be that sister chromatid phase. So remember, we're aiming to get to the anaphase part where those chromatids are pulled apart. So DNA polymerase has to make sure those nucleotides are linked properly via those covalent bonds between the sugar and the phosphate molecules. Replication is called semi-conservative. It means that one side of the DNA molecule will be from the original strand when we get our two new ones, and one of them will be from newly synthesized nucleotides, essentially. So here's our new section built by the red nucleotides, and our old stuff carries on through. So even though we have two new strands, they're sort of half old, half new. Now, the template strand that was used to start with, so this blue one here is the template strand, and the red nucleotides being built on again, you're going to end up with two identical double strands there, because this is going to end up being identical to this one here and this to this one. This is a nice BioNinja picture. All right, some more really good BioNinja information. Here's a great diagram. Before we knew how this replication worked, there were three hypotheses that could have been proposed for the method of replication of DNA. So number one, the conservative model. It talked about having an entirely new molecule synthesized from the DNA template, and so this is all new stuff. The second model, the semi-conservative model, which we now understand is the correct one, said that, okay, we're going to split that and have half old and half new, so semi-conservative, half conservative. The third one was called dispersive. It meant that the new molecules of DNA are made from segments of both the old and the new all the way through. Now, we didn't really know which one was right, and so Messelson and Stahl were able to experimentally test the validity of these three models using radioactive isotopes of nitrogen. Now, nitrogen can be heavy or light. It can have a few more neutrons in there or a few lighter. So they used nitrogen 15 and nitrogen 14 to do that, and we know that nitrogen actually is involved in all of those nucleotide base pairs in the DNA molecules. So what they did was they prepared DNA molecules using the heavier stuff, so the 15, the nitrogen 15, and then they only let them replicate in the presence of nitrogen 14. So they could actually track where the 15 nitrogen was at the end of it. So what they then found after one replication was that they could see, and we can have a look if we zoom in on here. I don't know if we can zoom in, there we go. The first replication here, it ended up having different DNA molecules with a mixture of the 14 and the 15. So straight away, that's knocked out the conservative model, because in the conservative model, we're assuming that some of the new DNA is completely brand new and the old stuff sticks together. Now in that in the second replication, I'll zoom out, pause, try that. In our second replication, what they found was some of the DNA molecules were still that mixture of 14 and 15 nitrogen, but then there were also some that were completely only made of the 14 nitrogen, which is the stuff they gave them to allow them to replicate. So that knocks out, once again, that dispersive model, because the dispersive model assumes that it's going to be a combination of all the new DNA molecules are a combination of the older ones. All right, PCR is called polymerase chain reaction, and it's kind of an outside the body and artificial method of replicating DNA, so without using its usual practices that are considered natural. So again, 3 nice simple steps, and the first step is denaturation. So it's tearing that DNA molecule apart at very high temperatures. And then the next step is called annealing. So what they do is they stick a little primer, which is a DNA marker, to kind of flag to the enzymes coming up to help that this is the bit we want you to replicate, just this bit and nothing else. The third step is, so during the annealing step, they actually cool it down to do that, to anneal the primer, and then they heat it back up again for the elongation. And they get an enzyme called Taq polymerase to come along and add in those extra DNA bases in the same way that DNA polymerase would do during the natural process. Now, Taq polymerase is a really interesting enzyme because it comes from bacteria in hot springs, which means it's really heat tolerant, so it can do its job at 72 degrees. And the name PCR, or polymerase chain reaction, it's a chain reaction as one is replicated, the next bit can continue and continue and continue. And it's also that polymerase, so it's adding, it's creating those polymers of DNA. So you only need tiny quantities of DNA to do PCR, and you amplify it really, really quickly. So we're talking each batch will double. So we're going from 1 to 2 to 4 to 8 and so on and so on. The only problem with that is if you drop a tiny fleck of dead skin into a sample for PCR, it's going to start amplifying your DNA, and that's not what you want. Now PCR can be used for many different things. You'll often see it on CSI because it's used in forensics quite often. If you can take a small amount of DNA at a crime scene, amplify it up so you can actually test it a little better. Used in medicine quite a bit, obviously. It can diagnose certain gene mutations, and it can diagnose different bacterial and viral illnesses as well with more certainty. And you're also amplifying genes in certain research studies to actually figure out what their use is and how we can manipulate that. All right, so there are our dot points. Once again, a reminder that these are only half of topic 2.7. The rest are on transcription translation next week.